Hi, hi everyone. Welcome. Today we have Heather Snell, who completed her PhD at the University of North Texas, combining cellular biology with pharmacology in the laboratory of Eric Gonzalez. Then she joined the laboratory of Kamran Khodaha at Albert Einstein College of Medicine in New York, where she focuses on understanding the receptors and channels underlying the intrinsic activity of Purkinje cells, specifically in disorders that affect the cerebellum, such as episodic ataxia. In this disorder, what's interesting is that patients display relatively few and mild symptoms for long periods of time, but then stress triggers the symptoms. Heather discovered that with stress, release of norepinephrine leads to disruption of Purkinje cells, which is then followed by a motor attack. In January 2024, she established her own research program at the neuroscience department at Yale School of Medicine, and where she is thriving. She's been awarded a fellowship from the Ford Foundation, from the Brain and Behavior Research Foundation, and she was also named Next Generation Leader by the Allen Institute. Great to have you.
Thank you so much. Thank you so much for inviting me to give a talk in this amazing series that I've watched so many of the YouTubes of. So I'm really excited to be included in that. And I'm really excited to give you a little glimpse of the work that I did in my postdoc looking at caffeine-induced attacks, but then also a little bit, if we get there, time-wise, of some of the work that we're doing in my lab that we've established over the past year. So we're going to talk about these disorders called episodic ataxia type 2 that are caused by mutations in the CACNA1A gene. And so let's just get right in. Let's see. We know as a field that the cerebellum has been more than just motor coordination, and that's been separated into kind of a map of the cerebellum where regions are more generally concerned with motor, but then other regions are responsible for language and social interaction. And what's kind of amazing is that the cerebellum is so highly implicated in autism spectrum disorder and a lot of these other cognitive disorders, just symptoms such as social communication, repetitive movements. And 80% of patients with autism spectrum disorder and ADHD actually have some kind of motor impairment. And we haven't really understood either the cerebellar contribution to the motor impairment in autism, but also the cerebellar contribution to the cognitive aspects of autism. And so my lab at Yale is really concerned with understanding the cerebellar contribution to both the motor and the cognitive symptoms in these cognitive disorders. And we're kind of looking at this through the lens of CACNA1A mutations. And so CACNA1A it encodes for a voltage-gated calcium channel. It's called the PQ type calcium channel. Specifically, it's this pore-forming part of the channel. So there's auxiliary subunits called alpha 2 delta and beta that make a functional channel. But the pore-forming subunit is really what is really important for passing calcium through this channel. And this channel, the P in PQ actually stands for Purkinje cells because it's so highly expressed in the cerebellum. This blue line is the cerebellum here, and then this is the rest of the brain. And so what's really unique about it is that throughout the CNS, these voltage-gated calcium channels are responsible for the release of neurotransmitters. So you have an action potential propagate, you have opening of these voltage-gated calcium channels, and you have the release of neurotransmitters presynaptically. Really, the only place that it's expressed postsynaptically is on Purkinje cells in the cerebellum, where you have a voltage event, you have opening of these PQ-type calcium channels that causes the activation of these calcium-activated potassium channels, and this is going to be the topic of the rest of my talk is kind of manipulating these SK channels to alleviate symptoms in our mouse models. And that causes the efflux of potassium. And these channels have been shown to be very important in the after hyperpolarization of Purkinje cells and the maintenance of their intrinsic activity. And so it's no surprise that patients with CACNA1A mutations, and this is a picture from the CACNA1A family meeting a couple of summers ago. I'm right there in the back. It was an amazing meeting that combined patients with CACNA1A mutations, their families, and researchers of CACNA1A. It's no surprise that majority of these patients have cerebellar symptoms, so cerebellar atrophy, ataxia, eye movements, nystagmus, But a subset of patients also have autism spectrum. About 25% of these patients have some cognitive impairment. And so this is a really good mutation and a really good genetic handle to start to understand how could the cerebellar contribution to the motor aspects, but also to the cognitive aspects, seeing as how this channel is so highly expressed in the cerebellum on Purkinje cells. But today we're only going to talk about the motor aspects of the mutations that cause episodic ataxia type 2, which is the second on this list of ataxias. So we've known as a field that the intrinsic activity of Purkinje cells, the maintenance of this intrinsic activity of Purkinje cells is really important. And that's what is underlying a lot of the ataxias and dystonias that we've seen tied to cerebellar disorders. And so multiple reviews, multiple papers have been and kind of investigated this both in Purkinje cells and in the deep cerebellar nuclei onto which they synapse. And our lab, in my postdoc lab, has recorded from many different models of ataxias and dystonias. Here is a lot of models that you probably are similar or are familiar with, like SCAWs, spinal cerebellar ataxias. This is a Friedreich's ataxia model at the end. And we found that this irregularity of firing, this ISICV, which is the standard deviation of the ISI over the mean. So it's just, oh, sorry, I think that's on a timer. So it's just the spaces, how regular the spaces in between each one of these action potentials is. And as you can see in a wild type, it's relatively regular. And in a transgenic model, this is our model of episodic ataxia type 2 called a tottering mouse. They will have these spaces and these clumpings. And so this ISICV is higher, and that increased ISICV correlates really well with the increased disability score, 0 being normal ambulation and 5 being these sustained dystonic postures. And so you can see this wild type here, this is our transgenic, is this orange dot here. And then a mouse during an attack is this highly irregular firing of Purkinje cells, and that correlates with this increased ISICV and increased disability score. And so then the lab was really focused on if we can decrease this ISICV, so make the Purkinje cells fire more regularly, can we decrease the symptoms of the patients and decrease this disability score? And so our model that we're going to be using today, the first model that I'm going to tell you about, is called the tottering mouse. And so each one of these rectangles is a mutation in the CAPNA1A gene that has been shown to cause episodic ataxia type 2. It's the most common form of episodic ataxias. And the mouse model, it's called the tottering mouse. It's called the tottering mouse because it totters. This is a spontaneous mutation that was found in Jackson Labs in the 70s. And these mice, similar to patients, when they are either physically stressed, physical or emotional stress, when they drink coffee or when they drink alcohol, it goes from this mild baseline ataxia to this long dystonic or dyskinesia that can last from a few hours to a few days. And if you physically restrain this mouse, or if you inject caffeine or inject alcohol, this mouse goes from mild baseline ataxia to these sustained dystonic postures that can last from an hour to an hour and a half. And so this is a really good model to start to understand what is the mechanisms behind these triggers, and can we add drugs to alleviate them to possibly give to patients. And so, as I told you, these triggers are things that we see every day. I think you all can agree we've been under a lot of stress. I've probably been in this 40% of employees that have cried from stress this year. It's been a very stressful year. And we tend to self-medicate when we're stressed. This is me at GRC this past summer at a chocolate factory. And also we drink a lot of coffee, so caffeine. And then we tend to go to happy hours. So this is a text with another new PI here where we meet with at happy hours. And so we have stress, we have caffeine, and we have alcohol. And patients are pretty much told to just stay away from stress, stay away from alcohol, and stay away from caffeine, which is physically impossible to stay away from stress. And the other two are kind of what make life happy. So we wanted to try to figure out what is the mechanisms behind these triggers so we can help alleviate patient symptoms. And we had some clues from previous work. So previous work in the Kodak lab, my postdoc lab, had shown that SK channels, so that channel that receives the calcium from the PQ type calcium channel. If you add an positive allosteric modulator, so activate these channels, you can alleviate some of the baseline ataxia in this mouse model. And so in this experiment, they conducted the rotarod test where you just put mouse on a rotarod, accelerating rotarod, and you see how long it takes them to fall off. So that's the latency to fall. And what they found is after they added chlorzoxazone into the drinking water of these mice, they increased in their ability to stay on the rotarod longer, not up to wild type levels, which is the black, but they did increase. And then when they stopped putting it into their drinking water, they came back down to baseline levels. And so we thought, okay, well, maybe there's some SK channel activity that we can manipulate that further causes increased motor impairment and causes increased Purkinje cell irregularity. And so the hypothesis was that all these triggers, so stress, caffeine, and ethanol, further inhibit SK channels. So in episodic ataxia type 2, you have about a 50% decrease in the amount of calcium coming in through PQ type calcium channels. So less activation of SK channels. So you activate them, the baseline ataxia gets a little better. The hypothesis was that somehow these triggers further decrease SK channel activity, causing increased irregularity of Purkinje cells and increased motor impairment. And so the first kind of tackle that I'm going to briefly talk about, because this has already been published, was looking at stress. We wanted to look at stress first because that is the most debilitating for patients. You can't really not be stressed. And so we got some clues from previous work from Alan Hess's group that showed that when you block noradrenergic pathway, you can actually prevent episodic dysfunction in episodic ataxia type 2, which was great. We also got some clues from previous work by John Edelman's group that showed that norepinephrine can reduce SK2 channel activity already through this kinase called CK2. This is casein kinase 2. It's constitutively active and it phosphorylates SK channels, decreasing their affinity for calcium and decreasing their activity. And this goes through alpha-1 adrenergic receptors. And so to test this, we did a lot of work. This was a very amazing collaboration with Ariel Vintenzone, who was a student and postdoc in Cameron's lab, and now has his own preclinical startup called MiriFizz, which if you're interested in preclinical work, please reach out to him. It's located in North Carolina. And Ezra Tara, who was a student and postdoc in the lab. And so I'm just giving you a brief overview of this, and what we showed was when you stress the mouse out, and our stress paradigm is restraint stress in a 50 ml conical tube for 10 minutes, and you blocked alpha-1 adrenergic receptors, you can prevent or significantly reduce the amount of attacks that these mice had as a response to stress. And when you injected norepinephrine alone independent of stress, you can induce attacks, suggesting this isn't indeed going through noradrenergic pathway. And then when you block noradrenergic pathway, alpha-1 adrenergic receptors, you can prevent those attacks. And then looking at irregularity, so again, this is our measure of irregularity of Purkinje cells. We did slice electrophysiology, extracellular, to record Purkinje cell activity, and we saw that norepinephrine alone significantly increased that irregularity compared to baseline. And when we added prazosin prior to adding norepinephrine, we could prevent that increase in irregularity, suggesting that indeed stress goes through alpha-1 adrenergic receptors. This was also shown by a Melanie Marks group recently that is going through alpha-1D receptors on Purkinje cells to cause these attacks. But what about CK2? So when we, to investigate CK2, one of the many ways that we did this was we knocked it down in the cerebellum. So we had an AAV against CK2, injected it into the cerebellum, and knocked down CK2. It has two catalytic subunits, alpha and beta, and we knocked down both. And when we knocked down CK2, what we found was kind of, we were surprised, but a happy surprise, is that when we knocked down CK2, we were able to prevent both stress and caffeine-induced attacks. This was great because this suggests that maybe, sorry, I think everything's on a timer, that maybe they're going through similar mechanisms, going through, somehow converging on CK2. And indeed, norepinephrine has been shown or caffeine has been shown to modulate the release of norepinephrine. So we thought, okay, maybe caffeine's also going through alpha-1 adrenergic receptors. This would be great. We'd have one pathway to rule them all. We'd be able to manipulate alpha-1 adrenergic receptors and prevent both stress and caffeine-induced attacks. As is science, it's not always the way that you think it's going to go. So to test this, similar to our experiments with prazosin, where we injected prazosin prior to our stress paradigm, we did this also. We injected prazosin, which blocks alpha-1 adrenergic receptors, and injected caffeine to see if it could prevent caffeine-induced attacks. And as I showed you before, prazosin prevented stress-induced attacks, but it did not prevent caffeine-induced attacks. We also tried a beta adrenergic blocker, propranolol, and that failed to prevent caffeine-induced attacks. So this suggests that caffeine going through CK2, but has a different upstream mechanism that is independent of alpha-1 adrenergic receptors. So as we often do with science, we have to pivot. We have to go back to the drawing board and figure out what could be the mechanism for caffeine. What does caffeine do? What is its main function in the CNS? And caffeine's main function is it blocks adenosine receptors. So that's what makes its stimulant activity, because when it blocks adenosine receptors, it causes the release of presynaptic glutamate, of presynaptic neurotransmitters that cause us to be stimulated and that cause us to kind of have this really great effect of caffeine. We also wanted to see which ones are highly expressed in the cerebellum, and A1 and a little bit of A3, but mainly A1 adenosine receptors are highly expressed in the cerebellum. So we kind of wanted to test this to see, independent of caffeine, if we block A1 adenosine receptors, can we cause attacks, right? Independent of caffeine. And then that's exactly what we did. And this is work from that was conducted by a group of us. And what we did is we injected these blockers of A1 adenosine receptors and one blocker of A3 adenosine receptors to see if A3 was also involved. And when we blocked A1 adenosine receptors, we saw that we were able to increase significantly the percent of attacks that these tottering mice had, independent of any trigger, so independent of stress or caffeine. And when we injected A3 adenosine receptors, we did not see a significant increase. So this suggests that caffeine could be going through the block of A1 adenosine receptors to result in this these caffeine-induced attacks. But what about Purkinje cell activity? And so the next question we wanted to ask ourselves was, is there an increase in Purkinje cell pacemaking with adenosine receptors and with caffeine, right? And is this going through A1 adenosine receptors if we actually do see this increase in Purkinje cell regularity? So again, we went to our slice electrophysiology. So we slice cerebellar slices from wild types and tottering mice. We record extracellularly because we're looking at calcium dynamics and we don't want to dialyze the cells, dialyze out the natural calcium buffers that the Purkinje cells have. And we can also record in the presence of synaptic blockers to isolate the intrinsic activity of Purkinje cells. So we had picrotoxin, CGP for GABA A and GABA B, and then we had CNQX or NBQX for AMPA and kinase receptors. And then we can add our caffeine, it's 100 micromolar, or norepinephrine in the case of stress to see how that affects Purkinje cell activity. And so when we did this, and I'm just going to show you the norepinephrine as a kind of reference, what we saw was that norepinephrine, this is wild types on the left, caused a significant increase in the irregularity of firing at baseline of tottering or mutant Purkinje cells. And then when we added caffeine in tottering mouse, we also saw that there was a significant increase in the irregularity of firing of the Purkinje cells with that application, suggesting that yes, this does go through the increase in Purkinje cell irregularity, which makes sense when we look back at that graph. We were looking at increased irregularity and increased motor dysfunction. And so again, we wanted to confirm that prazosin is not causing this or is saving the Purkinje cells, preventing this increase in irregularity like we see with wild type or like we see with norepinephrine. So when we add prazosin with norepinephrine, again, we see this prevention of this increase in irregularity that we see with norepinephrine alone. But when we added prazosin with caffeine, we did not see a significant decrease in this irregularity that was caused by caffeine, suggesting again, it's not going through alpha-1 adrenergic receptors. So now we know that norepinephrine is going through alpha-1 adrenergic receptors. Caffeine, and what I haven't shown you is we also did test when we blocked A1 adenosine receptors in slices, we saw an increase in the irregularity of firing. The hypothesis is that caffeine is going through presynaptic release of glutamate from parallel fibers onto Purkinje cells, right? And we wanted to kind of go deeper into this because this was well established before we started that norepinephrine can increase CK2 activity through alpha-1 adrenergic receptors. But we did not know what the connection could be between A1 adenosine receptors and CK2, right? But we did hypothesize that we see this increase in release of glutamate after we have the block of A1 adenosine receptors, and that it could be interacting with mGlu1 because in our slice preparation, we blocked AMPA and kainate receptors. And so we thought, okay, we're not blocking mGlu1s. Let's see if it's going through mGlu1 with the hypothesis that if we block mGlu1s, we should be able to prevent caffeine-induced attacks and caffeine-induced irregularity if they're required for the propagation of this mechanism. And so that's exactly what we did. We got an mGlu1 antagonist, which is called J&J. It has a bunch of numbers after it, but I'm just going to call it J&J for this talk. And we bath applied it similarly to what we did with prazosin. And when we did this, we saw a significant reduction in this increase in ISICV that we see with caffeine alone, suggesting that mGlu1s are required for this increase in irregularity. We also went in vivo. Ariel also did this experiment in vivo, and similar to what we did with prazosin, we injected this J&J, a blocker of mGlu1s, prior to injecting caffeine, and we wanted to see what is the percent of attacks that these mice had. This is one of the most robust experiments that I've ever seen. Increasing concentrations of J&J completely blocked caffeine-induced attacks. And so these mice are very happy. They do not have attacks. And so this suggests that mGlu1s are required for caffeine-induced attacks and caffeine-induced Purkinje cell irregularity. And so this was really great because now we have kind of this mechanism by which caffeine releases glutamate released through the block of A1 adenosine receptors on parallel fibers and interacts with mGlu1s to activate some pathway. We still had no idea what the kind of connection between mGlu1s and CK2 could be. We already had this established pathway for stress, but we really need to establish how mGlu1 activity could be causing an increase in CK2 activity, this kinase that decreases SK channel activity and causes these attacks. And so it's been shown that CK2 can actually interact and regulate other glutamate receptors, so AMPA and kainate receptors and MDA receptors. And so we thought, okay, maybe mGlu1s are physically coupled to CK2 and that their increase in activity just naturally increases CK2 activity or increases CK2 phosphorylation of SK channels. And so to test this, we went to some biochemical and biophysical assays. We did two things. So first we did a co-IP, a co-immunoprecipitation with mGlu1 and CK2 to see if they are physically coupled in wild types. Do they come down together? And so this is an assay where we can add an antibody for one of these proteins, and then we can blot for the second one to see if that came down with the other protein. We also did some immunohistochemistry for phosphocalmodulin. Phosphocalmodulin being a reader out of CK2 activity because it phosphorylates the calmodulin on SK channels, decreasing their affinity for calcium. And so we use this phosphocalmodulin as a readout for CK2 activity. And so we wanted to see if we blocked mGlu1s, can we caused a decrease in this amount of phosphocalamodulin in Purkinje cells. And so we have three groups. We had baseline. We had a group where we injected J&J, this blocker of mGlu1, prior to caffeine injection. And then we had a group where we just did caffeine injection, and then we sacrificed them during the attack to see what is the amount of phosphocalamodulin in these animals. And so for the co-IP, we first blotted for mGlu1s because that was the best antibody that we had. And we got robust amounts of mGlu1s when we pulled mGlu1s down. And then we blotted for CK2 after we pulled mGlu1s down. And we got a band, very bright light band, but we got a band for mGlu1 or for CK2 when we brought mGlu1s down, suggesting that they could have some kind of physical interaction. We also tested for mGlu5 because mGlu5 are also expressed in Purkinje cells in the cerebellum. And so we did not see a band for CK2 when we blotted or I did our experiments for mGlu5, suggesting that this could be something specific to mGlu1s. For our immunohistochemistry, we probably can't see it very well in these images, but we performed immunohistochemistry for tagging for phosphocalamodulin in our cerebellar slices from tottering mice, either no attack, during caffeine, or when we added J&J before caffeine, when we injected that into the mice. And what we saw was during no attack, this is all normalized to no attack, we saw a slight about 13% increase in the amount of phosphocalamodulin in Purkinje cells during an attack or during caffeine attack. That was not seen when we added J&J prior to the attack, suggesting that mGlu1s are required for this increase in phosphocalamodulin that we think is going through this CK2. And so now we kind of have our mechanism for caffeine, which is going through blocking of A1 adenosine receptors and promoting the increase in release of glutamate. We also did some fiber photometry experiments to show that caffeine causes this bulk release of glutamate in the cerebellum that's interacting with metabrotropic glutamate receptors that are physically coupled to CK2, and that when you have this increase in mGlu1 activity, you have this increase in CK2 activity, and that further decreases SK channel activity, causing these motor attacks for caffeine. And so both of these converge on this CK2 activity, which is possibly something that we could use as a therapeutic handle to help treat these patients and prevent these caffeine and stress-induced attacks. And so that was our next goal, was to see if we could find a drug that we might be able to reasonably use in a short amount of time to get to patients to actually prevent these attacks. And so we found this drug, CX-4945. I can't say the other name. I'm horrible at saying it. It is a potent and select inhibitor of CK2. A lot of these, there's multiple kinases, so CK1, CK3. This is very selective for CK2. It's orally bioavailable, meaning that it's something that patients would just be able to take a pill, and they'd be able to possibly have the alleviation or prevention of their stress or caffeine-induced attacks. Again, it inhibits CK2, and it has been approved as an orphan drug by the FDA to treat multiple different cancers. It's now in a clinical trial to treat COVID symptoms. And so this is kind of a hot topic drug that many people are trying to use for multiple different disorders. So we wanted to see if it would be viable to prevent caffeine and stress-induced attacks in episodic ataxia type 2. And so the first thing that we did was we went to our slice preparations, and we found that when we bath applied CX-4945, similar to what we saw with prazosin and similar to what we saw with J&J, we were able to prevent norepinephrine-induced irregularity, and we were able to prevent caffeine-induced irregularity in our Purkinje cells in the slices from the tottering mice. And so this suggests that indeed this could be used to prevent caffeine and stress-induced attacks. We also gavaged our mice because it's orally bioavailable. We could gavage the mice with this drug. We did it for 3 consecutive days to have it build up in the CNS. And then we stressed the mice, or we injected caffeine. And when we did this, and these are two separate cohorts, we were able to see that we significantly reduced the percent of attacks these mice had with stress and with both caffeine. And then after we let it wash out for about 2 days, they were back to having their attacks. And so this is something that the patients would be able to take daily as a prophylactic for potentially having these attacks. And so this was really exciting. We were really kind of one of the goals now that I've started my own lab is to see if this kind of CK2 pathway, SK channel pathway, is generalizable to other CACNA1A mutations and other episodic ataxia type 2 mutations, specifically patient mutations. The tottering mouse is a great model, but it's not a patient mutation. No patient has the mutation that the tottering mouse has. It was a spontaneous mutation. And so in my postdoc, I worked with the CACNA1A Foundation to make a mutation in the CACNA1A gene that is more representative of patient symptoms. And so can this SK channel serve as a convergence point or a potential therapeutic handle for cerebellar disorders? And so the mutation that we made is carried by a few patients that have been identified by the CACNA1A Foundation. And it's called D1634N. They have motor impairment, but they also have multiple cognitive symptoms. So this is getting at kind of what I talked about at the beginning, where we wanted to use these models to kind of understand the cerebellar contribution to motor, but also the cerebellar contribution to cognitive dysfunction as well. And so this is a D to an N, aspartic acid to an asparagine. So just literally a acid to a neutral shift. Now we have done work to show that it is actually a loss of function mutation, so a decrease in the amount of calcium coming through the channel by this mutation. Patients are described as slightly ataxic. Their parents describe them as clumsy, so they have these possible intermittent ataxia bouts. They have impaired communication, so speech difficulties, slow auditory processing of language, poor response to environmental sound despite normal hearing. Seizures are very common among CACNA1A mutation patients with CACNA1A mutations, and these patients do have seizures. They also have poor working memory, mild facial dysmorphia, and social relatedness early on was a strength, but progressed to decrease in social interaction, and poor attention and emotional regulation. So we have this wide variety of phenotypic heterogeneity in these patients, and we really wanted to use this mutation as a way to kind of look at that. And so this mutation is in a very highly conserved area of the CACNA1A channel. And this has given us a lot of benefit because we can look at this mutation across many different models. And so we're looking at it in mouse here. We also have a collaborator, Perry Kirshen, who is looking at this in C. elegans. And we also now have a collaborator here at Yale who is looking at this in Drosophila because it is also conserved. And so we can start to understand the mechanisms shared across many different models to kind of add power to the different mechanisms that we're looking at and kind of finding in this mutation. And so the first thing we wanted to look at was, does this mouse, so we used CRISPR to knock in the mutation into a mouse at the homologous site, which is D1585N. And we wanted to characterize any kind of motor impairment that this mouse might display. These are heterozygote mice, so they have a healthy allele and they have a mutated allele. The homozygote mice are viable, but they don't live past about P21 because they are very ataxic. And so we are working on getting some more of those to survive so we can start looking at what a full knockout would look at, or a full knock-in of this mutation would present as. But these are similar to patients, they're heterozygotes, so it's actually a really good model to compare to patient symptoms. And so when we put them on the catwalk, what we see is they do have motor, kind of a gait impairment. This is the wild type gait, and on the bottom is the heterozygote gait for this mutation. They are slower, so they have a decrease in velocity, but they also have a decrease in the regularity index of their gait. So how normal is their gait process? We also notice that they tend to have a more, and we don't really know the meaning behind this, but they tend to have more of an impairment on their right sides than their left side. And so we are still kind of investigating that, but it appears that they kind of, if you look at the kind of the heat map of their feet when they walk on this gate or this catwalk, they almost walk on their toes instead of walking on their full kind of palms. And so we're working on kind of characterizing this. I have a student running these experiments again to increase the ins, and we'd really like to understand what is the gait differences in these mice. These are adult mice, and to see if this is something that is developmental. Are they acquiring this gait because they have motor impairment? And if the drugs that we give them, this SK channel activators, these CK2 blockers, will that alleviate these gait impairments? We also see that they do have motor deficits, so we've run them on both the balance beam and the rotor rod. And in the balance beam, we see that these mice have an increase in the number of foot slips, significant increase, especially with the smaller beam on day 2. And we also have run them on a multiple days in accelerating rotor rod, and we see that there is a significant decrease in the amount of time that they can stay on the rotor rod when they meet this plateau, which happens around day 5. And so I showed you earlier that when they added chlorzoxazone to the tottering mice, they were able to alleviate some of the motor impairments when they activate these SK channels. So we wanted to see if this is true in this animal model as well. And so what we did is we added chlorzoxazone to their drinking water at day 16, and we assessed them on the rotor rod throughout for about 13 days. And then we washed out, and this is work that I did in my postdoc, and unfortunately Christmas happened, so I had to cut it off. I didn't have enough time to go out, but what we did see is that when they started drinking chlorzoxazone, they did increase their motor ability to stay on this rotor rod longer. And then when we washed it out of their system, they came back down to baseline rather quickly. So this suggests that indeed their baseline ataxia or baseline motor impairment could be due to manipulation of SK channels. They also show some stress-induced phenotypes. We also have tested them for caffeine. They also show some caffeine-induced phenotypes. They do not appear similar to the episodic ataxia, which is very much an hour-long bouts of these motor impairment. They're more sporadic and they're more mild in nature, but this is the mouse at wild time or at baseline. You really wouldn't be able to tell by eye that it has any kind of motor impairment. And then we kind of see two things, which we're working on collaborations for the first one. What we see is that we think they're having seizures because they have a lot of these bouts of immobility in the corners. We think they're having some seizures, and we want to work on quantifying that and really characterizing that. But we also see this increased motor impairment on the bottom, and they're leaning. And so they do lean to the right, which could explain kind of this rightward kind of impairment that we're seeing when we do the catwalk. But they do have these bouts of increased motor impairment after they've been stressed. And we are working on quantifying that. And then the question is, can we give CX-4945 to prevent these bouts of motor impairment and possibly the seizure propagation in this mouse model? Sorry. We have also recorded from these animals. So we went back to our electrophysiology or slice electrophysiology. And what we find is that baseline, these mice do display a significant increase in their regularity of firing of their Purkinje cells at baseline. And they also tend to have a decrease in their average firing rate, which is something we don't see in the tottering mice, but we only really see in this animal model. And so then we wanted to say, I showed you earlier that chlorzoxazone can alleviate some of the motor impairment, and that is through a decrease in this ISICV. So it goes from a higher ISICV, more irregular firing, to more regular firing in the presence of chlorzoxazone. And so we wanted to see if bath application of chlorzoxazone can also alleviate the irregularity of firing of Purkinje cells in this animal model. And indeed, what we found was when we bath applied chlorzoxazone in our heterozygote slices, we saw that we can significantly decrease the irregularity of firing, but it does not alleviate the average firing, which we think that this might be synaptic. So PQ type calcium channels are also on parallel fibers, on granule cells. And so this could be a change in possibly a decrease in release of glutamate from parallel fibers in this animal model that is might not be in the tottering mouse. And so we do want to kind of pursue this with some photometry to see if there is a change in the release of glutamate from parallel fibers, or try to understand this that's not coming from the intrinsic activity of Purkinje cells, but might be more of a synaptic phenomenon. And so just to summarize everything that I told you, I know I told you a bunch of things. So caffeine and stress kind of converge on this SK channel dysfunction. So block of CK2 prevents both attacks and the Purkinje cell regularity caused by caffeine and stress, possibly providing a molecular handle to start to understand some therapeutics that can prevent these attacks in patients with EA2, and possibly other disorders that have stress and caffeine-induced attacks. This might be we're hypothesized that perhaps decreased SK channel activity at baseline could be the requirement to kind of propagate into these caffeine and stress-induced attacks. And we see that in a lot of different cerebellar disorders, this irregularity of firing or SK channel activity contribution. And so we would love to explore other channelopathies that involve SK channel activity and Purkinje cell regularity to see if this could be a conserved mechanism. In our mouse model that we've created, this D1585 in mouse, we've shown that we can recapitulate the motor symptoms of patients. So we have baseline ataxia, we have some motor impairment that is very transient from stress, and we also see that with caffeine. And we also showed that motor impairment and irregularity are alleviated when we activate SK channels, similar to the tottering mouse, which is kind of queuing up all of our further experiments that we want to do in the lab, which is a lot of things. We want to see if we can manipulate SK channel or CK2 in this mouse model. Is this a conserved? I do want to really understand that CK2 and GLORA1 interaction further, and how is that alleviating or contributing to motor symptoms. mGLORA1 is highly implicated in a lot of other ataxias and dystonias, and so could this interaction be contribute to those other kind of cerebellar disorders? We have started to characterize the cognitive symptoms in these models, so novel object recognition, social novelty, and aggression. And we also want to characterize epilepsy in this model to see if it's cerebellar in origin and if we can help alleviate those symptoms also. And we also have a Purkinje cell specific knock-in of this model that we are characterizing, and we hope that we can actually understand what is the Purkinje cell specific contribution to this disorder. And so with that, I would like to thank my postdoc lab, Cameron Kotica, and everyone else, Ariel Vintonzone and Ezra Tara, and everyone else who contributed to the work, and also my lab at Yale, which is steadily growing, and then all the behavior cores at Yale that we would not be able to do this work without, and my funding sources. And thank you for listening.
Well, thank you. Thank you, Heather. Thank you for that wonderful presentation. And if anybody has questions, please go ahead and raise your hand. I'd be happy to call on you. Maybe I can start by asking you a little bit about, I thought it was surprising that the caffeine-induced effect on regularity was via a completely different pathway, parallel fiber glutamate release, as compared to the norepinephrine pathway on the Purkinje. But yet both of these pathways, they seem to produce increased coefficient of variation on the spike time regularity. How is I can understand the effect of norepinephrine, but how would caffeine produce the same irregularity?
Yeah, so we think the irregularity kind of sits in SK channel dysfunction, right? And so the hypothesis is that caffeine it goes through this increased mGluR1 activity. And the mGluR1, we don't really understand the interaction between mGluR1 and CK2 yet, but we know that there is some physical interaction and that when we block mGluR1s, we can decrease the activity of CK2 through that phosphocalmodulin assay. And so we think that increased mGluR1 activity causes an increase in CK2 activity, which that further decreases SK channel activity. So we think both of them, both caffeine and stress, through different upstream mechanisms, converge on increasing CK2 activity, which when that happens, it decreases SK channel activity, which will change that after hyperpolarization of the action potential and lead to burst firing of Purkinje cells.
Let's see. Yes.
Does that answer your question?
Nicholas, yes, you did, Heather. Thank you.
Okay.
Nicholas, go ahead. You have your hand up.
I see the hand.
I'm trying to, can you hear me well?
Yeah.
Yes. Hi.
Okay, cool. Uh, I've been involved in some research on caffeine, and my understanding was that the caffeine has impact on vascular reactivity. Do you find any effect of SK channels on vascular reactivity?
We haven't looked at vascular reactivity, but that would be, do you know what concentration of caffeine you're looking at?
That was in humans. So within physiology from
Okay.
like 50
Okay. So
to
it's not like
500
yeah, so it's
milligrams.
not the
No.
high concentration that causes ER
Yeah.
release of caffeine.
Hmm.
We haven't looked at vascular reactivity, but I think vascularization in the cerebellum and looking at that might be something that might be contributing, but we haven't looked at that at all. So that can be something that we do look at. I haven't looked at that in that context, but thank you. Yeah, we haven't looked at it. Oops,
Thank
sorry.
you.
Let me write that down. I see one more hand.
Hi, I was curious if you see any cell loss in any of these
Mm.
conditions, and whether or not that interacts in any way with these
Mhm.
changes in baseline physiology of Purkinje cells. I guess dovetailing with that, if that does occur, is there any sort of progression of any kind in any of these disorders, or is it typically more static?
Yeah, so we don't really see, so we're looking at that now in our newer mouse model. We're doing a lot of IHC to see if we see any Purkinje cell loss, especially in specific Purkinje cell populations that might be more vulnerable. Because I know in a lot of cerebellar disorders, you see certain Purkinje cell populations like zebrin or posterior versus anterior tend to be more vulnerable to loss. We haven't seen, so in the tottering mouse, you don't really see, and in a lot of CACNA1A, in patients, you do see cerebellar atrophy, but that's normally in the white matter. It's not really in Purkinje cell counts. We haven't seen any Purkinje cell. We've done up to 9 months in our CACNA1A model. We haven't seen any Purkinje cell degeneration. And in the tottering mouse, what's actually really interesting is you don't really see any degeneration until really late in the disorder. And then you actually see that the attacks get better because we do know that sick Purkinje cells are worse than no Purkinje cells. And that's been shown by the Purkinje cell specific mutation model, PCD model, that if you get rid of all the Purkinje cells, the motor impairment actually gets a little better. But we haven't seen any gross loss of Purkinje cells yet. Maybe later in the disease propagation, but we've gone up to 9 months in the CACNA1A model that I showed you, and we haven't seen any Purkinje cell loss. We are looking at morphology changes, so to see if there's any changes in the dendritic arborization, but we're looking at that now. Yeah.
Harsh,
Very cool, thank you.
Mhm.
Harsh, please go ahead. You have your hand up.
Yes. Thank you. Thank you so much. Dr. Snell, this was an amazing talk. Thank you.
Thank
Really
you.
enjoyed it. Two quick questions. One for you probably mentioned this, but I missed this. In the co-IP experiments, when you
Mhm.
pull out mGlor1 and you say CK2, was there a way to isolate Purkinje cells or like some type of cell type specificity, or is it just bulk cerebellar tissue?
It's
Um, eh,
bulk. Yeah,
okay,
unfortunately, it's bulk. But we're working on trying to pull out Purkinje cells. We have a L7. So we're doing some like turbo ID right now where we have L7 turbo ID, which we can bring out Purkinje cells only. So then we can start to
sure.
only look at Purkinje cells. But yeah, it might be overcome with granule cells. But yeah, it was just bulk
Okay.
cerebellar lysates.
Fair enough. And then the second question was, you said that the CK2 does modulate other glutamatergic receptors too, but
Mm-hmm.
is it actually known that this physical interaction exists with other glutamatergic receptors, especially like NMDA
Um,
receptors? Is it known that?
So with NMDA receptors, I think they've shown that there's a physical interaction. Most of it has just been chemical.
Okay.
So when you decrease activity, this is what happens to the NMDA receptors.
Hmm.
Some work has been done in cell cultures where they have done some co-IPs in cell cultures and shown that in cultured neurons that you can get a CK2 and I think. I think it was also NMDA receptors. A lot of work has been done mostly on NMDA receptors and CK2 in the hippocampus.
Okay.
And because it's highly implicated in like Alzheimer's disease,
Sure.
and Parkinson's disease, I think, but oh no, Alzheimer's disease, they're using CK2 as that's also one of the preclinical trials that they've done with an Alzheimer's disease. But most of it has been kind of biophysical, chemical, not so
Hmm.
much kind of this physical interaction. Yeah.
Okay, cool. Thank you.
Hmm.
Wonderful. Any other questions for Heather? Heather, that was amazing. Thank you so much.
Thank you so much. Thank you for having me.
Wonderful, really wonderful. Learned a lot.
Thank you.
Bye, everyone.